Picture a conference room in the early 2000s. On one wall, a printout stretches from floor to ceiling, a genetic map of humanity built from over 1,000 DNA samples collected on every inhabited continent. A researcher steps back from it, expecting to see the tidy boundaries textbooks had described for over a century. Black, white, Asian, neatly separated blocks of biology. Instead, the map in front of them is a blur of overlapping gradients with no clean edges anywhere. Someone in the room says out loud what everyone is starting to realize: the boxes were never really there. Somewhere in a lab, a geneticist lines up thousands of genomes on a screen, samples pulled from every inhabited continent on Earth. The plan is simple: compare them, sort them, see how cleanly humanity splits into the racial categories most of us grew up believing in. What comes back on the screen isn't clean at all. It doesn't sort. It doesn't split. And the actual result is stranger and more revealing than almost anyone expects. Stay with me, because by the end of this video, the way you think about race and DNA is going to change completely. Let's start with the study that really opened this up. In 2002, a team led by geneticist Noah Rosenberg analyzed DNA from over 1,000 people across 52 populations worldwide, part of the Human Genome Diversity Project, a massive international effort to sample genetic variation across humanity as broadly as possible. The goal was straightforward: feed the data into a computer algorithm with no instructions about race, ethnicity, or geography, and see what clusters emerge purely from the genetic patterns themselves. The results made headlines around the world, and they still get cited constantly in genetics classrooms today, because what the algorithm found didn't map onto the racial categories most societies use at all. Here's the first big surprise. When researchers actually measured genetic variation across humanity, the overwhelming majority of it, somewhere around 85 to 90%, depending on the specific study, exists within populations, not between them. In plain terms, two random people from the same so-called racial group are often more genetically different from each other than the average difference between two large population groups. This finding traces back to population geneticist Richard Lewontin, who ran the numbers in 1972 and found that if every race on Earth vanished except one, you would still retain the vast majority of human genetic diversity within that single group. Race, as a biological sorting tool, barely captures anything about the actual pattern of human genetic variation. So if genetic variation doesn't split cleanly by race, what does it actually look like? The honest answer is that human genetic variation is clinal, meaning it changes gradually across geographic distance, the way temperature changes gradually as you travel from the equator toward the poles. There's no sharp line where one race ends and another begins. Instead, allele frequencies, the rates at which specific genetic variants show up in a population, shift slowly and continuously as you move from one region to a neighboring one. A person's DNA in, say, northern India looks progressively more similar to populations in Iran as you move west, and progressively more similar to populations in Southeast Asia as you move east, with no clean boundary anywhere along that gradient. Genetic maps of humanity look far more like a smooth, continuous gradient than a set of separate, boxed-in categories. So if genetic variation doesn't split cleanly by race, what does it actually look like? The honest answer is that human genetic variation is clinal, meaning it changes gradually across geographic distance, the way temperature changes gradually as you travel from the equator toward the poles. There's no sharp line where one race ends and another begins. Instead, allele frequencies, the rates at which specific genetic variants show up in a population, shift slowly and continuously as you move from one region to a neighboring one. A person's DNA in, say, northern India looks progressively more similar to populations in Iran as you move west, and progressively more similar to populations in Southeast Asia as you move east, with no clean boundary anywhere along that gradient. Genetic maps of humanity look far more like a smooth, continuous gradient than a set of separate, boxed-in categories. Imagine walking on foot from Norway down through Europe, across the Middle East, and into East Africa, stopping in every village along the way to sample a person's DNA. At no single point on that walk would you cross a line where one population's genome suddenly, dramatically shifts into a completely different one. Each village looks genetically almost identical to its immediate neighbors. It's only once you've covered the entire distance and compare the two endpoints side by side that a large difference becomes obvious, and that difference is the accumulation of thousands of tiny, gradual shifts along the way, not a single hard boundary anywhere on the route. This is precisely why trying to draw firm racial lines across human genetic variation always ends up feeling arbitrary. Any line you draw could just as easily be drawn 100 miles to the left or right, because the underlying biology never gave you a natural place to put it. This is actually where Rosenberg's 2002 study gets more interesting, not less. The researchers weren't naive about this Clinal reality, so they specifically tested what happens when you force a clustering algorithm to sort people into a fixed number of genetic groups, say 5 clusters, and see how those clusters line up against geography. When they did this, 5 broad clusters did roughly correspond to major geographic regions: Sub-Saharan Africa, Europe, and the Middle East, and Central and South Asia. Together, East Asia, Oceania, and the Americas. But here's the catch that got lost in a lot of the popular coverage at the time. These clusters only appeared because the researchers told the algorithm to produce a specific number of groups. Ask the same algorithm for 3 clusters, or 10 or 20, and you get entirely different groupings, because the underlying data isn't naturally divided into discrete categories to begin with. It's continuous variation that can be sliced into however many bins you choose, and the number and shape of those bins is a human decision, not a biological one written into the DNA itself. Rosenberg himself, along with several co-authors, published a follow-up paper in 2005 specifically to address the confusion this caused. They stated plainly that the clusters identified by the algorithm should not be interpreted as evidence for the existence of a small number of discrete, biologically fixed human races. The clustering was a reflection of geographic sampling patterns and historical migration and isolation, not proof of clean, natural racial divisions. In other words, the very researchers whose work is most often cited to argue that genetics confirms race is real were the same researchers who explicitly, repeatedly, and publicly said that's a misreading of their own results. It's worth understanding where the old racial categories actually came from in the first place, because they weren't discovered in nature, they were invented, and fairly recently in the scope of human history. The 5-race system most Western societies inherited traces largely back to Johann Friedrich. Blumenbach, a German naturalist working in the late 1700s, who sorted human skulls into categories he labeled Caucasian, Mongolian, Ethiopian, American, and Malay, based largely on skull shape and, by his own account, aesthetic judgment about which skull he personally found most pleasing as the supposed ideal form. Blumenbach never had access to DNA, migration data, or population genetics of any kind. He was working entirely from a small collection of skulls and the assumptions of his era. And yet a framework built on that foundation shaped scientific, legal, and social thinking about human biology for the next 2 centuries, right up through the census categories, medical intake forms, and everyday assumptions many of us grew up with. Let's talk about why this matters beyond the academic argument, because there's a real historical weight behind this question. For centuries, race was treated as a rigid biological fact, something written into blood and bone used to justify slavery, colonialism, forced sterilization, and immigration. restrictions across multiple continents. 19th and early 20th century scientists spent enormous energy trying to prove that racial categories corresponded to measurable biological differences in intelligence, temperament, and worth, research that has since been thoroughly discredited both on ethical grounds and on the actual data. Modern genomics didn't just fail to rescue that old framework, it dismantled it from the inside using the exact tools, DNA sequencing and population genetics, that some had hoped would finally provide the hard biological proof those old theories were missing. Now here's where the science gets specific enough to actually explain the confusion. If race isn't a clean genetic category, why do people from certain regions look so visibly different from each other? The answer is that a small number of highly visible traits, skin pigmentation being the biggest one, evolved independently and relatively recently in human history, largely in response to how much ultraviolet radiation a population's ancestors were exposed to. Populations that lived closer to the equator, with intense year-round sun, evolved darker skin to protect against UV damage and folate degradation. Populations that migrated to higher latitudes with weaker and more seasonal sunlight evolved lighter skin over time because lighter skin allows more vitamin D synthesis in low-light environments, which was critical for survival in regions where UV exposure was limited for much of the year. Here's the part that surprises almost everyone. Skin pigmentation genes are controlled by a relatively small number of genetic variants, and they evolved recently and rapidly compared to the rest of the human genome, meaning skin color changes fast, in evolutionary terms, without dragging the rest of a population's genome along with it. This is why two populations can have nearly identical skin tones, while being genetically quite distant from each other elsewhere in the genome, and why two populations with very different skin tones can be genetically closer to each other than either is to a population that merely looks similar to them. Skin color, in other words, is a poor proxy for overall genetic relatedness. It's one adaptive trait shaped by one specific environmental pressure layered on top of a genome that tells a much more complicated and much older story. A landmark study published in Science in 2017, led by geneticist Sarah Tishkoff, sequenced the genomes of over 1,800 people across Africa and found that the genetic basis for skin pigmentation is far older and more complex than previously assumed, with several of the genetic variants associated with lighter skin actually originating in Africa itself, tens of thousands of years before modern humans migrated out of the continent, and later spreading to other parts of the world through population movement and interbreeding. This upended a long-standing assumption that light skin evolved exclusively outside Africa after humans left. The study found that some populations within Africa, including certain San communities in southern Africa and groups in East Africa, carry notably lighter skin tones than many other African populations, driven by some of these same ancient genetic variants, a detail that gets completely lost when an entire continent, home to more genetic and phenotypic diversity than anywhere else on Earth, gets treated as a single, uniform racial category in everyday conversation. It turns out the raw genetic material for a wide range of skin tones, from very dark to comparatively light, already existed within African populations long before any large-scale migration out of the continent took place, which is part of why Africa today holds more human genetic diversity in total than the rest of the world combined. That last point deserves its own moment because it's one of the most consistently underappreciated facts in all of population genetics. Africa is not just one region among several roughly equal ones. Because anatomically modern humans originated in Africa and lived there for the vast majority of our species' history, before any group migrated elsewhere, African populations have had far more time to accumulate genetic diversity. Every population outside Africa descends from a relatively small subset of people who left the continent in one or more migration waves tens of thousands of years ago, carrying only a fraction of the genetic diversity that existed back home. This is sometimes called the serial founder effect, and it was mapped with striking precision in a 2005 study led by geneticist Sohini Ramachandran, which measured genetic diversity across 52 populations worldwide and found a near-perfect statistical relationship. The further a population's ancestors had traveled by the most likely ancient land and coastal routes from East Africa, the lower that population's genetic diversity turned out to be, almost like a genetic odometer recording the distance of an ancient journey. Each migration further from Africa involved a smaller, less diverse subset of the previous population's genetic variation. A single village in parts of sub-Saharan Africa can contain more genetic diversity than the entire population of Europe or East Asia combined. So if the old racial categories don't map onto real genetic clusters, what does modern genetics actually reveal instead? Population geneticists today generally talk about ancestry, not race, because ancestry can be mapped with real precision using specific genetic markers tied to particular migration histories, while race remains a social and historical category that was invented to sort people, not discovered in their DNA. A person's ancestry can be traced with genuine scientific rigor to specific regions, migration events, and historical populations, the kind of research we've covered on this channel before. But layering the label race on top of that ancestry data adds nothing scientifically. It imports old social categories onto a biological reality that never actually organized itself that way to begin with. There's a particularly striking piece of evidence for this that comes from looking at populations that don't fit neatly into any conventional racial category at all. Take populations in the Horn of Africa, parts of Ethiopia and Somalia, where genetic studies have found ancestry components that blend deep African lineages with genetic contributions from populations historically associated with the Middle East, reflecting thousands of years of movement and contact across the Red Sea. Or take populations in South Asia, where genome-wide studies led by researchers including David Reich and Kumarasamy Thangaraj have identified two major ancestral components, one related to populations further west toward Iran, and one representing a deeply diverged ancient South Asian lineage found nowhere else on Earth, blended together in different proportions across nearly the entire subcontinent. Neither of these populations fits cleanly into any of the broad racial boxes commonly used in everyday language, and that's not an exception to the rule. Populations like these are common once you actually start looking closely at genome-wide data from around the world, which is exactly the point. The boxes were never a good fit to begin with. Genetically isolated populations make this even clearer. Ashkenazi Jewish populations, for instance, descend from a relatively small founding group that lived in Central and Eastern Europe for many centuries, largely marrying within the community due to social and religious separation from surrounding populations. Genetic studies show Ashkenazi Jewish populations carry a distinctive blend of Middle Eastern and European ancestry, a direct genetic record of a specific historical migration, and centuries of relative isolation that doesn't map cleanly onto either a Middle Eastern or European racial box because it's genuinely both, fused through a very specific and well-documented history. Finland offers another case. Finnish populations, despite looking, by conventional categories, similar to their Scandinavian neighbors, actually carry a genetically distinct signature shaped by a small founding population and centuries of relative isolation in the north, which is part of why Finland has become such a valuable resource for medical genetics research, since rare disease-linked variants that are hard to study in large, mixed populations show up far more clearly in a population with this kind of deep, documented isolation. Neither of these groups exists because of race. They exist because of specific, traceable history, which is exactly the kind of story broad racial categories are too blunt to capture. There's also a lot of confusion caused, ironically, by the popularity of consumer DNA testing itself. When people take an at-home ancestry test and get back a pie chart labeled with percentages next to country or regional names, it's easy to read that as scientific confirmation of racial categories. It isn't. Those percentages are generated by comparing your genome against reference panels built from modern populations with documented roots in specific regions. And the results describe ancestry, meaning where segments of your DNA most closely match people living in a particular place today, not membership in a race. Two people who both test as, say, 70% one broad category and 30% another can have arrived at nearly identical percentages through completely different family histories, different specific ancestors, different migration paths, compressed into numbers that look deceptively simple on a screen. The tests are a genuinely useful tool for mapping ancestry. They were never designed and don't function as a scientific instrument for confirming race. This is also where a lot of the confusion around medicine and race comes from, and it's worth addressing directly because it has real consequences. For decades, some areas of medicine used broad racial categories as a rough proxy for genetic risk, assuming, for example, that a patient's self-identified race could reliably predict things like drug metabolism or disease risk. Modern genomic medicine has moved steadily away from this approach because self-identified race turns out to be a weak and often misleading predictor of the specific genetic variants that actually matter for a given health condition. Two patients who identify with the same racial category can carry completely different versions of a gene relevant to, say, drug metabolism, while two patients from different racial categories might share the exact same version. Researchers now increasingly favor looking at direct genetic ancestry, or better yet, sequencing the specific genes relevant to a given condition, rather than relying on race as a genetic stand-in, precisely because the underlying biology doesn't sort itself by the old categories. Sickle cell disease is probably the clearest example of how this kind of mislabeling happens. It's often described as a condition that affects Black people, and it does occur at high rates in many African and African-descended populations, but the actual genetic reason has nothing to do with race as a category. The sickle cell trait persisted at high frequency in specific regions because carrying one copy of the mutation provides real protection against malaria. So in any population that lived for generations in a region with high malaria exposure, the trait became common through natural selection, regardless of what continent that population happened to live on. This is exactly why sickle cell trait also shows up at meaningfully high rates in parts of the Mediterranean, the Middle East, and India, regions with a long history of malaria exposure and no direct genetic relation to sub-Saharan African populations at all. The pattern tracks malaria exposure, not race. Framing sickle cell as a Black disease isn't just imprecise. It has led to real documented cases of doctors overlooking the condition in patients who don't fit the assumed racial profile and over-testing or misdiagnosing patients who do, simply because the geography of a mosquito-borne disease got mislabeled as the geography of a race. None of this means human populations are genetically identical or that ancestry doesn't matter. It clearly does, and we've spent plenty of time on this channel exploring exactly how specific traits trace back to specific ancient populations and migration events. What the science actually shows is more precise and, honestly, more interesting than the old racial framework ever was. Instead of a handful of broad fixed categories, human genetic history looks like an intricate web of migration, isolation, mixture, and adaptation, unfolding differently for nearly every population on Earth, shaped by specific environments, specific journeys, and specific historical events. Reducing that entire tapestry down to a handful of racial boxes doesn't just oversimplify the science, it actively erases most of what actually happened. It's also worth being clear about what modern genetics does not support, because pseudoscientific claims about race have unfortunately persisted well past the point where the underlying data debunked them. There is no genetic basis for claims that any racial group is inherently more intelligent, more athletic, or more capable than another as a biological class. Traits like intelligence and athletic performance are influenced by enormous numbers of genes interacting with environment, upbringing, nutrition, and opportunity in ways that make any simple, race-based genetic explanation scientifically indefensible. Serious geneticists across essentially the entire field reject these claims, not out of political caution, but because the actual genomic data doesn't support them and never has. This is a point worth dwelling on for a moment, because even researchers who study human genetic ancestry in serious depth have gone out of their way to draw a hard line here. David Reich, one of the most prolific population geneticists working today, and someone whose research this video has referenced multiple times, wrote publicly in 2018 specifically to distinguish between two very different claims. The first, that human populations have real, measurable, average genetic differences shaped by distinct migration and adaptation histories, which his own research demonstrates repeatedly, and which this video has spent a lot of time exploring. And the second, the far more serious and unsupported claim that those differences translate into a clean hierarchy of traits like intelligence mapped onto old racial categories. Reich was explicit that the science supports the first claim and does not support the second, and that conflating the two is exactly the kind of misuse of legitimate genetic research that responsible geneticists have to actively push back against. Ancestry is real and traceable. Racial hierarchy is not, and pointing to genetics to justify one has never been backed by the actual data, no matter how many times that argument gets recycled. Since Rosenberg's original 2002 study, the scale of this research has only grown, and the conclusion has only gotten stronger, not weaker. The 1000 Genomes Project, launched in 2008, eventually sequenced full genomes from over 2,600 people across 26 populations worldwide, an enormous leap in resolution compared to earlier studies. More recent efforts, including the Human Pangenome Reference Consortium, have pushed even further, explicitly working to build genetic reference data that better represents global diversity, instead of relying so heavily on genomes of primarily European ancestry, which dominated earlier genomic databases and skewed a lot of early medical genetics research toward one relatively narrow slice of humanity. Every expansion of this dataset, every added population, every added genome has continued to reinforce the same underlying pattern Rosenberg's team first documented: continuous overlapping variation shaped by migration and geography, not clean division into a small number of fixed biological races. So let's come back to where we started. When researchers actually compare the DNA of people across the globe, expecting to find humanity split into clean genetic teams, they don't. They find something closer to a single sprawling, deeply interconnected family tree, one where every population is a cousin of every other population, more closely related to some, more distantly related to others, but never cleanly divided into the small number of boxes so much of human history assumed were biologically real. The actual result of comparing DNA across every population on Earth isn't that race is real and hiding in the genome, waiting to be confirmed. It's that the genome tells an entirely different, far more interconnected story, one that never matched the boxes we built on top of it. That's the real shock hiding in this research, and it's a bigger one than most people expect walking into this topic. Not that science proved old assumptions about race correct, but that it proved, repeatedly, rigorously, and from multiple independent angles, that those assumptions were never an accurate map of human biology to begin with. The deeper you look into the actual genetic data, the less humanity looks like a set of separate boxes, and the more it looks like exactly what the fossil and archaeological record has been telling us all along. One connected, endlessly migrating, endlessly mixing species with a genetic history far richer and far stranger than the simple categories most of us were taught to take for granted. Go back to that conference room for a second, to that researcher standing in front of a wall-sized map with no clean edges anywhere on it. That moment wasn't a failure of the research. It was the research working exactly as it should, revealing what was actually there, instead of confirming what everyone expected to find. Every genome added since has only sharpened that same picture. Not humanity sorted into a handful of separate rooms, but one continuous house, with every population connected to every other by some hallway of shared ancestry, some closer, some further, but none of them ever truly separate. If this changed how you think about DNA, race, and ancestry, like this video and subscribe, because we dig into what the actual science says every single week, not the popular myths. Drop a comment with what surprised you most, or which population's genetic history you want us to cover next. And if you know someone who still thinks of race as a hard biological fact, send this their way. The data has been telling a different story for over 2 decades now, and most people have simply never heard it laid out this clearly. This